Okay, we're back with sample A. The only reason I stopped there was because the recordings get too long and YouTube can't cope with them, basically. So, okay, so we have two samples, and what we would do is we would take some of our sample A, and we would isolate total DNA. Lots of different ways you can do that. Don't worry about it too much. Lots of ways we can do that. But isolate total DNA. And then I would do a PCR on the total genomic DNA that I got from the poop sample. And bear in mind, that's going to contain everything. It's going to contain eukaryotic cells from the gut, any parasite cells that might be in there, but also all the bacterial cells. So all of that is in there. Is what I would then do is I would pick a gene which is highly conserved between the members of the group of organisms I'm interested in. So in terms of bacteria, I would pick the 16S ribosomal DNA gene. And I picked that because it's conserved between members of the group, but there's enough differences that I can use the sequence information that I'm going to get to determine what the individual species would be. So if I've got species 1, 2, 3, 4, and 5, there's enough difference between the 16S ribosomal DNA genes in those species that I can distinguish one species from another. Why do I use 16S ribosomal DNA? Well, because it's unique to bacteria. Humans don't have 16S ribosomal DNA. They have different ribosomal DNA genes. So all I'm going for are the bacterial samples, the bacterial DNAs here. So what I would do is, if we think about all our DNA, these are all our pieces of DNA. Okay, here are all our pieces of DNA. Okay, and there are all different kinds of pieces of DNA here. Okay, and some of them are going to be pieces of genomic DNA from the bacteria which have the 16S ribosomal DNA gene on it. So I add to my sample, I add PCR primers which recognize only the 16S ribosomal DNA gene. And so these are all kind of different pieces of DNA from my sample, different species of bacteria that my primers recognize. So then I do my PCR reaction, and all I would be amplifying are these pieces of DNA. All the other pieces of DNA get swamped out by the PCR of these pieces of DNA. Now, these are special PCR primers. So these PCR primers have little adapters on them. So I'm just going to draw them in here, and you'll see what adapters are for in a moment. And so these are going to be adapter sequences. So the primer that we use for our PCR has got a little extra sequence, piece of sequence on called an adapter. And there are actually two different types of these. I've just drawn them all in red, but we could draw a blue one and a red one. So there's two different types of these, one for the 5' end and one for the 3' end. And so when we do this, we would end up with a PCR fragment, which is maybe, 250 base pairs long. So let me clean this up a little bit, and we will keep going. So we've done that PCR, and so all that's been amplified out of the total DNA sample is this small piece of DNA, 250 base pairs long, which corresponds to the 16S ribosomal DNA gene. So that means in my little PCR tube, I've got all of the 16S ribosomal DNA fragments from all of the bacteria that are in that sample. Okay, so what I now do is I go to the first part of the sequencing reaction. And so to do that, you've got to imagine that you've got this chip, kind of like a microscope slide, I guess. And on that microscope slide are attached short pieces of DNA which are complementary to those adapters that we had on our PCR products. And I'm actually going to put in two different types here, just so you can see a little bit better how this works. So here would be all over the surface of this little chip, you'd have these different little pieces of DNA. So they're just kind of hanging out there in the breeze, kind of hanging around. We then take our PCR sample that we got from our stool, and we add that to our little chip here. So imagine that we've got a piece of DNA that has a PCR. We got a PCR fragment, and then we've got a piece of DNA that we amplified. and on the other end is the other piece of primer with the other adapter on it. So what happens is these pieces of DNA, when you add them to the slide, the little pieces of DNA in the adapters, which are complementary to the adapters on the slide, they anneal. And you get this little bridge between two parts of the slide. You then add a DNA polymerase, and that extends the adapter that we had to start with. That extends the adapter that was on the slide, so we get a second piece of DNA made. And you keep repeating this process. You can basically separate the DNA from the slide again, re-anneal it, and you keep doing these cycles over and over and over again in what is called bridge PCR. It's exactly the same principle as PCR. So what you end up with is, on the slide, you end up with these clusters, these little islands, if you like, of the pieces of DNA that you added. So that's a kind of a second PCR step, but it means that all of these clusters are all the same sequence. So it means we can make multiple sequencing runs of the same piece of DNA. So that's the first thing we do. And so I'm going to, again, back out of this, erase this. I'm sure there's a quicker way for me to do this erasing, but whatever. This is kind of fun for me. So, okay, so what's the next thing we've got to do? The next thing is we have to sequence those pieces of DNA. So that's kind of going to be the next challenge. So if we think about how a piece of sequencing might happen, is what we would have is, on our chip we've got the piece of DNA. Actually, let's draw this the other way around, because that's how I had it before, and that'll make more sense, I think, if we draw it the other way around. So this is our slide, right? And then here is our what colour did we use before? Did we use red? Let's use red again. Here is our adapter, which is attached to the slide, and then we've got our piece of DNA, and it's kind of hanging out in the breeze. Okay, so what we do is we add another little tiny primer in here that's complementary to our adapter sequence. So we add that in, and that's just called a universal primer. And then we add in, so let's just say that the first base here is an A, and then the next base is a C, and then a G, and then a T, and then a C, and then an A, and an A. So let's say that's the sequence we've got. So what we add in then is we add in a collection of 4 nucleotides. So we add in T, A, C, and G. So these are special nucleotides. So what will happen is that an enzyme, a polymerase that you add, will grab hold of this T and it will stick it in opposite the A. Now, what you expect to happen is then for the G to get added in here, and then the C to add in, and then an A, and then a G, and then a T, and you expect that to happen. Okay, that's not what happens. That's what you expect to happen, but it doesn't, because this little T here has been chemically altered so that you can't extend it. And so the 3' hydroxyl on that T has been chemically modified so that when the polymerase tries to add in the G opposite this T over here, the T cannot be extended. And so the polymerization stops. And this T is also fluorescent. And so that T, let's just make it fluorescent, let's make it red. Okay, so it's red. Okay, so the T is red if my pencil ever works. So the T is red. So that's fluorescent. So we shine our fluorescent lamp on it, and it fluoresces. And because it fluoresces red, the machine says, okay, the first base is a T. You then chemically treat the T so that you remove the blockage to extension on the 3' hydroxyl of the T. And so then you add another bunch of nucleotides, and then we can add in the G. And again, the G is blocked so that it cannot be extended. And once again, the G has a fluorescent tag on it, and let's make it kind of some kind of green. Yeah, that'll work fine. And so the G's got a fluorescent tag on it. Okay, so once again, we illuminate the G, and the G fluoresces, and the machine reads it and says, great, the next base is a G. Then we do the same thing over. We deprotect the G, we wash out all the old nucleotides, we add in new nucleotides, and the polymerase then adds in whatever the next base is. So in this case, we'd add in a C. And so you just go through this process of deprotecting the base that was just incorporated, or nucleotide that was just incorporated, and then you add in another nucleotide, and that can be added on to the deprotected nucleotide from the previous step, and you illuminate the sample, and whatever color you get fluoresced at this spot on the slide. Remember, this is all happening in one little cluster in a slide, and so you're recording the fluorescence in each of these little clusters. And so this means you can do thousands and thousands of sequences on one of these small little chips. And if you're interested in seeing one of these chips, go up and talk to Professor Miller, and he's got a chip in his office, and you can see what these look like. They basically are about, let's see if I can draw one kind of to scale so you get an idea of the scale this operates on. So this is happening on a slide which is literally about that big, and it's got a channel in this end, and then the channels branch off, and so each of these lanes in here, you've got thousands and thousands of places that you can actually do the sequencing reactions. So one of these chips can sequence thousands and thousands of samples. And that's how you do next-generation sequencing. This is one example. This is called Illumina or Solex sequencing. If you look on Wikipedia and you just go and you just look up next-generation sequencing, there are several types of this, but this is the type that's, I think, most common and most easily understood. So that's next-gen sequencing, and that's all I've got for you. I hope that's interesting, and I don't think you'll learn that in any other classes, so that's pretty cool. I will see you tomorrow.